The overall goal of this procedure is to image resident tumor-infiltrating CD8 T cells labeled with fluorescently coupled antibodies within human lung tumor slices using confocal microscopy.
This method can help answer key questions in tumor immunology, such as the nature of the elements that positively and negatively regulate T-cell migration within human tumors. The main advantage of this technique is that it allows us to monitor the dynamic behavior of resident CD8 T cells in a preserved human tumor environment.
We first had an idea for this method when we established a method in murine lymph nodes to track T cells plated onto slices, a protocol originally published in JOVI in 2011.
To begin, add 1 gram of low melting point agarose in a container, and then add 20 milliliters of sterile calcium and magnesium-free PBS. Microwave this solution to completely dissolve the agarose. Cool this 5% agarose solution in an incubator at 37 degrees Celsius for 5 minutes. In the tissue culture hood, add 1.1 milliliters of complete rPMI 1640 medium into each well of a 6-well cell culture plate. Then, using a pair of sterile forceps, place a sterile 30 mm cell culture insert into each well. Incubate the plate at 37 degrees Celsius for 30 minutes until later use. Next, place the tumor biopsy in a 10 cm plastic dish, and using a sharp sterile blade, cut the tumor into a small cube-shaped fragment that is approximately 5 by 5.